Okay, today we're going to talk about protein synthesis. There's two steps to protein synthesis: transcription and translation. Okay, here we go. Come on, slideshow. There we are. So I saw this online and I thought it was funny. This person says, just got an email from a prof. As a reminder, you are required to wear clothes during Zoom meetings. Rules are made when they become necessary, not before. Please wear clothes. All right, this module has 4 parts: DNA structure and DNA replication, which we've already done. Today we're doing protein synthesis, and then next time will be mutations. Let's do a little bit of review first. The directionality of DNA. Remember that a nucleotide has 3 parts. There's a phosphate group here, there's a sugar molecule here, and then there's the nitrogenous base over here. Then there's the nitrogenous base. Notice that the sugar molecules and the phosphate molecules, these guys, bond together to form the outside of the DNA molecule. We call that sugar phosphate backbone. And then the nucleic acids, which are on the inside, nitrogenous bases, which are on the inside, are bonded together with hydrogen bonds. And it's really important that these hydrogen bonds that are weak are in the middle here, because when it comes time for the cell to access the information that's stored in the DNA, we want it to be able to unzip that very easily so it can access that information. And then the strong bonds, the strong covalent bonds on the outsides of the DNA molecule will keep holding all the nucleotides together in the right order. Each one of these units is a nucleotide. Right here on that side, and then on the other side we've got some nucleotides as well. I just want you to notice that the nucleotides on one side of the molecule are all facing one direction with their phosphate pointed down. And on the other side of the molecule, they are facing the opposite direction with the phosphate pointed up. That's going to become relevant when we talk about the direction in which the DNA information is read and copied. And this has to do with the sugar molecule over here. Remember that at each corner, we assume that there's a carbon. And the carbons are numbered. So here we have the first carbon of the molecule. That's called the 1'. We have the 2', the 3', the 4', and the 5' carbon. I want you to notice that the hydrogen, or in this case, the OH group right there, comes off of the 3' carbon. And then the phosphate group comes off of the 5' carbon. So we call this the 3' end of the nucleotide, and that's the 5' end of the nucleotide. On the left side, you see that the 3' carbon right there is the last carbon of that end of that side of the DNA. So we call this the 3' end. And then up here, the 5' carbon is the last carbon. And so we call that the 5' end. And then it's exactly the opposite on the other side of DNA. The 3' end is the last on this side, and the 5' end is the last on this side. And because of this opposite nature of DNA, we call DNA anti-parallel, because there's 5' to 3', and then 3' to 5' on the other side. The information flows in opposite directions. If you need or want a little more of a review, you can watch this animation from last time. Moving on, our next part of review, structural differences in DNA versus RNA. There's 3 major structural differences. The first one is that in RNA, we have a different base, uracil. Uracil is used in place of thymine. So wherever you would see thymine, it's been replaced with uracil. Also remember that pyrimidines bond with purines. 1 carbon ring bonds with another that has 2 carbon rings. So T with A, C with G, except in uracil, we have U in the place of T. The second difference is that the sugar is different. In DNA, we have deoxyribose, and in RNA, the sugar is ribose. And the reason they are named this is because of what comes off of the 3' carbon. In ribose, there's oxygen and hydrogen, OH. And in deoxyribose, the oxygen has been removed, just the hydrogen. So deoxyribose, that's why it's called. Also, the perfect couple does exist, a purine and a pyrimidine. This is adenine and this is thymine. Adenine is my favorite. I'm not a nerd. Come on. All right, and the third difference is that the DNA molecule is made of 2 strands wrapped around each other like this, 2 helices. We call it a double helix. RNA only has 1 side. There's a single strand, so it's called a single helix. And this is because every RNA molecule is a copy of 1 side of the DNA. And the fact that DNA is single-stranded allows it to fold up into 3-dimensional shapes because nucleotides of the RNA are attracted to one another by their hydrogen bonds. And then that 3-dimensional RNA molecules, those can do various jobs around the cell. And most of them act as enzymes, which means they help speed up chemical reactions. Some of them are just structures that do other things. One of them we're going to look at today is tRNA. It's called short for transfer RNA. And these little teeth represent the nitrogenous bases, the A's, the U's, the C's, and the G's. And you can see that they're being attracted to one another with hydrogen bonds. That's what these little dotted lines represent. So that's caused the tRNA to fold up in a specific way. There we go. All right. Next, let's review the base pairing rules. Remember which bases go together on DNA. We have G with C and A with T. So if you were to go through here, you would find that across from C is G, E is A, G is C, A is T, C is G, and A pairs with T. Now, the bases are held together, remember, in the middle with hydrogen bonds. And at this point, in RNA, U replaces T. So if G goes with C and A goes with U, and let's change that to a U instead of a T, I want you to pause the video and go ahead and try and write down what you would find on the other side of this RNA molecule. Right, so you would have C pairs with G, U with A, G with C, A with U, C with G, and A with U again. So some of you might be thinking, but Professor Heckman, you just said that RNA is always single-stranded, and here you have a double strand of RNA. And that's true. But remember that sometimes RNA can coil up into these 3-dimensional shapes, and in that case, the bases might be attracted to one another through hydrogen bonds, as you can see in this tRNA. Right. Now we finally get to the part 3, the protein synthesis. We have 2 steps, transcription and translation. Today's question is, you are made of protein, so how do we get from this DNA, to you? And you may remember from last time, we talked about the central dogma of biology, and we'll just review this again real quick. So let's say you want to make some food. You have a delicious, tasty pizza recipe that a friend has. And the first you want to get a copy of the recipe. Well, why? Because you don't just want to take her recipe. You want her to have a copy as well. And if you have your own copy, you can take it anywhere you want. And then using that information on that new copy, you, the chef, you make the pizza. So it's basically just a transfer of information between different forms. And that's exactly what the central dogma is like. The central dogma of biology just shows the flow of information from DNA to RNA to protein. And a cell can do 2 things with its DNA. It can either use its existing DNA as a template to make more DNA. That's DNA replication. That's what we learned about last time. Or it can use its DNA to make proteins. And protein synthesis has 2 steps. The first one is transcription. right there, in which DNA is used to make RNA. Now, DNA is not turned into RNA. We're just making an RNA copy of the information in the DNA. And the second step is translation. And translation is where the information in the RNA is used to make a protein. My little pizza is covering up protein. It should say made of amino acids. So the RNA isn't turning into anything. It's not turning into protein, but the information is being used to make proteins. It's a transfer of information between these three forms. Here's some really important vocabulary. When a gene is active, when it's being used, we say it's being expressed. So for example, if you have hairy hands, then the gene for that trait is active. So it's being expressed. And if your body doesn't show that trait, if you don't have hairy hands, we know that either the gene isn't being expressed or it's possible that you just don't have it at all. Oh, let's go take a look at that gene real quick. Wait, just kidding. Let's not look at that one. We're going to look at another one later on. Remember each one of these, okay? So go ahead and watch RNAi Discovered to 139 and come back when you're done. All right, let's do a little bit of review again. Prokaryotic cells have a nucleoid area right there, and the eukaryotic cells, they have a nucleus, and they also have a nucleolus within the nucleus. And remember, the nucleolus is where the ribosomes are constructed. We're going to talk about ribosomes in a minute. Eukaryotic cells, their DNA is contained in the nucleus, and outside the nucleus, which is what fills the rest of the cell, is a fluid that's called the cytoplasm. Everything else in the cell floats in this fluid. Organelles, for example, the mitochondria, which you can see right here, these are all mitochondria, the endoplasmic reticulum, the Golgi body, and others. And most importantly for us, the ribosomes are located, the finished ribosomes are located in the cytoplasm. And so all that stuff is outside the nucleus. The DNA is separated from the rest of the cell by a membrane called the nuclear membrane that surrounds the DNA and it keeps it safe. And prokaryotic cells, they have DNA as well, but it just kind of floats around in this, this ambiguous area, and wherever the DNA is at the moment is called the nucleoid area. Remember, I also want you to remember that a DNA molecule can have hundreds of different genes on it on a single DNA molecule, and each one of those genes would code for a different protein. Here's a simple overview of the process of protein synthesis in a eukaryotic cell. Remember, a eukaryotic cell has a nucleus. You've got the DNA in the nucleus here. It's separated from the rest of the cell, from the rest of the cytoplasm, and the membrane that separates them is called the nuclear membrane. Sometimes it's called the nuclear envelope. And there are also openings in this membrane so that certain things can go in and out. And those structures are called nuclear pores. The first step in protein synthesis is to make an RNA copy of the genetic information on the DNA, and that process is called transcription. DNA isn't turned into RNA, it doesn't become RNA, the information in the DNA is just copied, it's transcribed into an RNA molecule. And then the second step of protein synthesis is where the information in the RNA molecule is read and a protein is created. RNA isn't turned into a protein, the information is just used, it's translated into a form of a protein. And the whole thing is just a transfer of information from one form to the other. Prokaryotic cells can also do protein synthesis, even though they don't have a nucleus, and they do it in much the same way, but they just don't have a nuclear membrane, keeping the DNA separated from the rest of the cell. So for the rest of our time, we're going to focus on eukaryotic cells, the ones with nuclei, not nucleuses. We've talked about cystic fibrosis before, and it's a really great example of the importance of the sequence of DNA bases in a gene. Once a protein has been created, it can actually go and do stuff. For example, there is one gene in your body for a protein, transport protein called the CFTR, and it creates a membrane transport protein, which, remember, those are the proteins that allow things in and out of cells. And this particular protein, the CFTR protein, assists in clearing mucus from the lungs. So here we have the DNA in the nucleus of the cell, and then through the process of transcription, an RNA copy is made, and the information goes out to the rest of the cell using that RNA copy. And then through the process of translation, through the process of translation, the RNA is translated into a protein form. And then that protein is free to go insert itself into the cell membrane and help clear mucus from the lungs. And in fact, we can go and take a look at that gene right now. It's super cool to have the entire gene like right here on the internet. So it's the CFTR. It's located on chromosome 7. Scroll down, there's a summary of what it does, which I'm not going to read. Shows all the little details, introns and exons. Down we go, and here is the gene itself. The origin is where it starts. And it just goes on and on and on. And this sequence of letters, remember, is telling you the order of the nitrogenous bases in the gene along the DNA. It's a really long protein, really long gene, really, really long. Anyway, back to our fabulous PowerPoint. There's one specific base which, if changed, it will cause the entire protein to cease functioning. And if the information in that, if that information changes, then the product is going to change as well. In this case, transcription and translation still happen. The DNA is still used to make mRNA, which is still used to make a protein, but the protein is wrong, and there's one amino acid is in there that's different, so it causes the protein to fold incorrectly into the wrong shape, and it can't do its job. And that means that mucus builds up in the lungs. There's breathing problems, scarring, all kinds of terrible things going on. That is just how important DNA is. Go ahead and watch this animation, and as you watch, answer what is the relationship between a chromosome, DNA, and genes. And then stop and do the critical thinking worksheet. Just part 1 though. All right. This figure is the same thing that we saw before with the central dogma of molecular biology, but it's in more detail this time. Here's our 2 parts again. We have DNA synthesis and we have protein synthesis. And in a cell, the DNA can be duplicated, and we call this process specifically replication or DNA synthesis. Just basically like putting DNA in a copy machine and coming out with an exact replica. Now we're going to move on to how to make proteins. Protein synthesis, it has 2 steps. The first is transcription, and the second is translation. Information about how to make proteins is stored in the DNA. And when the cell needs to make a protein, the information in the DNA is copied into a new form, RNA, right there. Unlike replication, this process only uses part of the DNA, only the specific part that the cell needs. In this case, in the image you can see that's only part of the double helix that is being used. It's not using the whole thing. And the RNA copy is coming out, and we're not turning DNA into RNA, just making a copy of the information. And here you can see this is the same RNA, and it's been straightened out. And this process is called transcription, and it's the flow of information from DNA to RNA. Then the RNA leaves the nucleus, and it goes out into the cytoplasm, just like this, where the information it carries is used to make a protein. And this process of using tRNA, or of using RNA to make proteins, is called translation. Go ahead and watch the demonstration. I've created a link for it on Canvas. And during the demonstration, you need to find the answers to the questions during the next 2 slides. These are really important points, and there's some of the major points of protein synthesis, and I'm not going to tell you the answers because they're right there in the video. Hint, hint, you might need them for the quiz. And then after the demonstration, do the critical thinking worksheet part 2. So here are the questions. Fill out the table as you watch the demonstration, and then answer those questions. And come back when you're done. And we're going to pause this recording. Hello? Hello? Oh my God. Pause. All right. Recording again. Cool. All right, so I want to talk a little bit more about how transcription begins. Transcription, remember, is the process of using DNA to create RNA. And the question is, how does RNA polymerase know where to start? So at the start and at the end of each gene, well, at the start of each gene is what I'm trying to say, is a special sequence of DNA bases. It's called a promoter, and I've put a little bracket around where that would be on this. Sometimes it's called a promoter region, and on this sequence, there's gathered a complex of several proteins right here. They're called transcription factors, and transcription factors are necessary for 2 reasons. First of all, before the process starts, RNA polymerase has just been running up and down the RNA, up and down the DNA, just been looking for a specific sequence of bases, and that sequence is the promoter. When it finds the promoter, it slows down, and when it finds its pile of transcription factors there, it's looking for that as well. And then when it slows down, it sits down on the promoter. But RNA polymerase is a very clumsy enzyme, and it needs a lot of help getting situated on the DNA and getting in just the right place and then staying there until it's ready to start. So once it's found this promoter and once it's found these transcription factors, then it binds to the DNA, and then more transcription factors and just come and just pile on. And finally, RNA polymerase can do its work. So I want you to notice that it's reading up from the 3' to the 5'. It's reading the template strand, and it's writing down from the 5' to the 3'. There, writing down from the 5' to the 3', just as DNA polymerase did when we were talking about DNA replication. So in that way, DNA polymerase and RNA polymerase are similar. Also notice that it's reading the template strand, which makes sense because when you're making something, you use a template. And because it's reading the template strand and it's matching bases to that strand, those bases are going to go in the same order as the other side of the DNA. So it will be an exact copy of the coding strand. It's not copying the template strand. It's copying the coding strand. It's reading the template strand to know what is on the coding strand. It's reading the template strand, which makes sense. And the coding strand is the one with the information we want, which is why it's called the coding strand. It codes for information in the protein. Right, promoter region, transcription factors, reads the template strand, and it creates an exact copy of the coding strand. So let's take a look again at this coding and anti-coding strand business. This coding and this coding and what am I talking about even? This is what isolation has done to me. Oh my God. Coding and template strands. Coding and template strands. So the RNA polymerase is reading this strand. That makes this strand the template strand, because it's using a template to make the mRNA. And you can see that it's matching up the RNA nucleotides according to the base pairing rules to make new RNA. We have G with C, we have A with T, we have C with G, and then U with A because we're using U and because it's RNA, and so on and so forth. And I want you to notice that the mRNA is not a copy of what's on this strand. It's a copy of what's on this strand. So here we have a G in the exact same place that there's a G on the coding strand. There's an A in the same place there's an A on the coding strand. C and C. T and U, of course, because we're in RNA. G with G. So we want the information on the coding strand because that's what's going to make sense to the cell when it's making this specific protein. So the coding strand is also called the sense strand. And if the RNA polymerase were going to make an exact copy of the information on the template strand, it would not get the information that it needs for the desired end protein. It would be the wrong information. It wouldn't make sense. So we call the template strand the antisense strand. Coding strand is sense strand, template strand is antisense. All right. Back we go. We got the promoter, we got the transcription factors. We're reading up and writing down. It reads the template strand and creates an exact copy of the coding strand. And how does it know where to stop? Well, at the end of each gene is a specific sequence called a termination sequence. Sometimes it's called a terminator, but usually a termination sequence. And I've put a bracket right here where it would be on this gene. And whenever the RNA polymerase reaches the termination sequence, it just loses its traction for the DNA and it sort of falls off, and then it releases the mRNA. And I also want to talk a little bit about mRNA processing. I'm going to stop this at this moment. Am I though? Am I really? My God. All right, continuing recording, sharing my screen, yeah, there we go. All right, I also want to talk a little bit about mRNA processing, which is also called mRNA modification. Remember that M stands for messenger because this RNA is carrying a message from the DNA to the ribosomes. So back in the DNA, the human genome contains about 25,000 genes, but it can produce about 100,000 different proteins. And that's way more proteins than there are genes. So how is this possible? Well, DNA is very efficient at storing information. On one strand of the DNA, there might be several different protein recipes just overlapping each other. So we've got, we've got our DNA right here. Let's say we want to make the protein melanin. First, we're going to make an mRNA copy of the entire DNA, which would be right here, coding strand, starting at the promoter, which would be over here, and ending at the termination sequence, which would be over here. But not all of that DNA is necessary for the melanin protein. There are parts of the message that aren't important to the final protein that we want, and those parts can be taken out. Those parts are called introns. They're called non-coding because they don't code for the final protein that we want at this point. You can see that the introns here are represented in a lighter pink color, and the other parts, the coding parts, are in dark pink. So once the introns are cut out, what's left of the RNA has to be spliced back together so that, again, it's one long continuous strand. The process is called splicing, and the segments that are left, the ones with the information that codes for melanin, those are called exons. And you can remember this with the phrase exons in, introns out, or you could say introns out, exons in. The thing is that this single DNA sequence, we've also done splicing, this single DNA sequence, so on the chromosome, it might have different information for several different proteins. And the way that you can get different proteins from the same DNA sequence is by making the same mRNA, but cutting out different parts of the message. We call this alternative splicing. The cell can splice together different exons and remove different introns in different places based on the protein it wants to make. And then, of course, remember, we always have the 5' cap and the poly A tail. Translation, it happens wherever ribosomes are found. Remember that translation is when mRNA is read and a polypeptide is made from the information in that mRNA. So transcription happens over here, creating the mRNA. The cell copies the information from the DNA into the mRNA, and then the mRNA is modified. And then after that, it leaves the nucleus and it will go to a ribosome, exits through the nuclear pore. It might find a ribosome floating free in the CNL, in the cell, in the cytoplasm. And then that translation would take place just floating free in the cytoplasm. Or sometimes it would attach, the mRNA would attach to a ribosome that is stuck to the outside of the rough endoplasmic reticulum. And when translation happens there, the ribosome just spins the new protein right down into the endoplasmic reticulum. Here you can see a picture of a ribosome, which is attached to the rough ER. We have the membrane here, the membrane of the rough endoplasmic reticulum. This is the inside. Here is the ribosome, and we've got the mRNA coming through. You can see here's the new polypeptide that's being made, just going right down into the rough ER. And it's already starting to fold into its final shape. And when it's done folding, then we can call it a protein. And inside the rough ER, the protein will be modified. Then it will be sent to the Golgi body, where it will have its final modifications. And remember, the Golgi body is called the post office of the cell, so to speak. So the protein will be further modified and then sent off to its final destination within the cell. All right, go back and watch the Central Dogma of Biology, this animation. And then after that, thinking back on the whole lecture, fill out this chart. And that will help you review for the quiz right here. And then that's it for today.